Hey everyone! So today, we're jumping into something that is completely changing how we fight cancer. It's called precision oncogenomics, and really, it's about a huge shift in thinking, from treating cancer with a sledgehammer to understanding that every single patient has a unique puzzle inside them that we can solve.
I mean, think about it for a second. What if every tumor came with its own instruction manual, a blueprint that spelled out all its strengths, all its weaknesses, and gave us the exact schematics on how to take it apart, piece by piece? That's really the core promise here. It's like we finally get to read the enemy's playbook before the game even starts.
For so long, the way we fought cancer was based entirely on where it was in the body. You had lung cancer, you got the standard lung cancer treatment. Breast cancer, you got the breast cancer drugs. It was very one-size-fits-all. But now, we're throwing that old map away. We're creating a completely personalized one for every single patient. Because we've realized it's not about the body part, it's about the tumor's unique genetic code.
And, this didn't just happen overnight. It's been a long journey. It really started back in the 70s, with scientists just noticing weird changes in chromosomes under a microscope. Then in the 80s, we started pinpointing specific genes that were causing trouble. The huge leap forward was in 2003 with the Human Genome Project. We finally had the whole book. And since about 2008, next-gen sequencing has given us the ability to read that book incredibly fast, making all of this possible in a clinic, not just a research lab.
Okay, so this whole idea of a personalized cancer map sounds amazing in theory, but what does it actually look like when you put it into practice? Let's dig into a real-world study that really put this idea to the test.
The study we're going to look at is called Genomes for Kids, and this was a truly landmark project. It took this super comprehensive genomic approach and applied it to one of the most difficult challenges in all of medicine, pediatric cancer.
The study looked at 309 young patients. Now, that's a really significant number, giving the researchers this incredibly rich set of data to start understanding the super complex world of childhood cancers. We have to remember, behind that number are real kids and young adults and their families. This research, it's not just about crunching data. It's about finding smarter, better, more precise ways to help them.
So, the researchers didn't just take one quick look. They used this really powerful three-pronged strategy. You can think of it like this. First, whole genome sequencing gave them the big picture satellite view of everything. Then, whole exome sequencing zoomed way in, giving them a super detailed street-level map. And finally, RNA sequencing acted like a real-time traffic report, showing them exactly which genes were active right at that moment.
So, what did all that digging turn up? Well, this is the headline number, and it's pretty mind-blowing. In a staggering 86% of these kids, this deep dive approach found clinically relevant information. That means things that could lead to a better diagnosis, a clearer outlook, a new drug to try, or even discovering a genetic risk. This isn't just theory. This is stuff doctors can use right away.
And that incredible 86% figure, it really drives home the most important idea in oncology today. Every single patient is unique. It means we have to stop treating cancer like a category and start treating it as a completely individual case.
This is what's known in the field as an N of 1 case study. It sounds technical, but it's a radical idea. It means the individual patient is the entire trial. Their cancer's genome is this unique puzzle, and the only goal is to find the specific pieces that are driving it, and more importantly, the ones you can target to shut it down.
And honestly, there's no better way to understand the power of this than through the story of Dr. Lucas Hartman. He was a physician who suddenly found himself on the other side of the equation, as the patient. And his words here, they just perfectly capture the hope behind this whole approach.
So let's just walk through his journey. Dr. Wortman relapsed with leukemia, and the standard chemotherapy just wasn't working. It was a really desperate situation. But then, his colleagues used RNA sequencing, that real-time traffic report we talked about, and found a gene called FLT3 was completely in overdrive. So they searched and found a drug already out there for other cancers that just so happened to target that specific gene. The result? A complete remission. That is N of 1 in action, and it saved his life.
Dr. Wortman's story is an absolutely incredible example of treating an existing cancer. But what if we could go even further? What if we could find cancer's footprint much, much earlier and then track it with a precision we've never had before? That's the future.
And this is where something called a liquid biopsy comes into play. Instead of a painful surgical biopsy, this is just a simple blood test. It works by hunting for these microscopic fragments of DNA that tumors actually shed into your bloodstream. It's kind of like finding a single tiny footprint in a massive forest that tells you exactly what you're looking for.
But here's the catch, right? The signal from that tumor DNA is incredibly faint. It's like a whisper in a hurricane. You have all this other normal DNA floating around, plus tiny errors that naturally happen during the sequencing process itself. So the whole challenge becomes how on earth you separate that one true whisper from all that deafening noise.
Well, the answer is this brilliant bit of digital error correction. See, before, the standard methods were just flooded with false alarms. But now, with a new technology, scientists add a unique molecular barcode, like a digital name tag, to each and every DNA fragment before they start analyzing it. That's the key. So now, if a random error pops up later, that new faulty copy won't have the original barcode, and the system just knows, nope, that's noise, and filters it out.
And the effect of this is just massive. This molecular barcoding reduces the number of false alarms, those misleading errors that look like real mutations, by 92%. That is a huge deal. It means we can finally trust the results we're seeing, turning a noisy, unreliable test into a high-fidelity cancer detection tool.
And having that kind of ultra-sensitivity just changes everything. We can now spot mutations that make up as little as 0.1% of the DNA in a blood sample. We can watch a treatment work in real time by seeing those signals fade away. And, the most exciting part, it opens the door to potentially finding cancer way, way earlier than we ever thought possible, long before it would ever show up on a scan.
So, we've gone from treating cancer with a blunt instrument to targeting it with a whole set of precision keys. We can map tumors, treat patients as the true individuals they are, and even find cancer's faintest traces in the blood, which really leaves us with the ultimate question. If every single cancer's code is unique, what's it going to take for us to finally crack them all?